Let's take a look at gene editing technologies. Successfully applied to genetic editing in plants are site-specific nucleases. These contain a DNA binding domain, that is, a domain which will bind to a DNA sequence of interest, and a nuclease enzyme. This nuclease enzyme is able to cut double-stranded breaks within the DNA. These gene editing technologies, which induce double-stranded breaks within the DNA, take advantage of the cell's natural DNA repair mechanisms. These DNA repair mechanisms repair the DNA, however, they leave it with a sequence which is different from the original. Let's first take a look at these DNA repair mechanisms. Let's take a look at how double-stranded DNA breaks are repaired by the cell. These DNA breaks may be induced by site-specific recombinases in the case of gene editing, or ionizing radiation, which can naturally cause DNA damage. Most DNA breaks are irregular, meaning that the breakpoint will occur in a slightly different position on each of the DNA strands within the double-stranded break. Irregular DNA breaks can leave what's called sticky ends, with overhanging single-stranded DNA bases on each of the broken ends of the double-stranded DNA. Let's see how the double-stranded breaks can be repaired by non-homologous end joining. Each of the broken DNA ends become bound by a set of proteins, the first of which is the Ku protein, which surrounds the double-stranded DNA. The second is DNA PKS, which binds to the Ku protein. DNA PKS then recruits Artemus and phosphorylates it. Artemus removes single-stranded overhanging DNA bases. Once this process has been completed on each of the broken ends, any overhanging single-stranded DNA bases will have been removed. On each of the broken ends, this forms what we call blunt ends, which are then to be ligated together, which means stuck together. Both of the blunt ends are then ligated together by the enzyme ligase 4, which is complexed with two other proteins called XXCCR4 and XXCCR4-like factor, otherwise known as XLF. Ligase 4 joins the two blunt ends together, and in this way the double-stranded DNA break has been repaired. However, due to the action of Artemis, any single-stranded DNA bases will have been removed. So once the DNA double-stranded break has been repaired, it is repaired to a sequence which differs from the original. The second way in which double-stranded DNA breaks can be repaired is by a process called homologous recombination.